Okay, well, let's get started. I appreciate everyone coming back for our fifth lecture. Today, I hope we'll be covering how to do cell growth and division in CompuCell. There are a couple of things I want to go over in addition to that, and we'll see how far we get. So I'd like to have people give very brief project updates, and I'd like to discuss how I'm going to try to formalize the project assignments going forward from this week. Maybe I should have done it 2 weeks ago since we had last week off. But I'd like to have people talk to you about that a bit. I'd like to talk to you a little bit about what is called practice of modeling, because it's important to help you think about how to present your projects and design your projects. And then I was thinking we might go over one of the old homework assignments from homework 3, although I may move that to the end so that we can be sure that we get through the cell growth and division. So the order of topics may change slightly. So one thing, I sent out a couple of emails about asking people to meet. Some people have been meeting with either TJ, Josh, Andy, or others, but some haven't. And for this particular semester, it's essential that everyone in the class meet with somebody, whether it's me, Andy, TJ, or Josh, on a weekly basis to get the projects done. I understand people are very independent, but these models are pretty complicated, and it's really critical that you have a weekly meeting with somebody to discuss your progress. So I'd ask people who didn't respond or who haven't been meeting with anybody so far that you please respond to that. So with that, I'd like to turn it over to you and say, get quick updates. I could certainly let you screen share if you want. You don't have to. Again, these updates could be quite short, maybe 2 minutes, 3 minutes. So maybe we'll start with Fanbo.
Yeah, sure. So basically, in the last week, I have already implemented a very simple widgets to display the mechanical, like using the Python widget, and I can share the possible simple demo if you want. So this is from an example of like the LC, the MC lattice, I think. So basically, you can play with this application. So first, this is a window which shows the simulation, the thread. And if you hit run, it will start running, like it will start to updating all the images based on the code you have, like for the simulation. And if you hit pause, it will just freeze all the scenes and stops all the scenes. And if you hit run again, it will just start, it will resume the simulation. You can also hit the reset button in order to reset the simulation. So if you hit a reset and you hit a run again, you can go back to the very beginning of the situation. So yeah. So in the following week, I think the thing I may do in this week or the next week is to apply the rotation of the camera of this. So you can using like the simple widgets to rotate the camera of the camera of the box to see it in different angles. Yeah, I think that's all I have for the updates.
Thank you very much. That's great. Kali?
So for my project, I will be working with Dr. Sugo. We had a meeting last week, and we decided on working with the paper by Moray and Hogeweg about how amoeboids self-organize into a fruiting body. And the first step of that is going to be trying to replicate the models in CompuCell, and then depending on how that goes, we may try to expand it a bit and then launch the final product as an app on NanoHub.
I was smiling because I turned on the closed captioning, and the closed captioning was displaying some amusing interpretations of the names. Sorry about that. Shouldn't be breaking. It shouldn't be breaking. I'm not breaking up because of what you're saying, but I can see the closed caption scroll. They were funny. But that's fine. Thank you. Tyler?
Yeah, let me show my screen really fast. Okay, so me and Dr. Sugo decided I'm going to do the early immune response of CD8 T cells. And a little background on what my paper is going to include is, so when an infection occurs, antigen-presenting cells will uptake the foreign antigen in the body, and via lymphatics, they'll make their way back to the lymph node. And these APC the APC cells will come in contact with CD8 T cells, and depending on the nature of the contact, it'll force the CD8 T cells to differentiate in different ways. And the model is a CompuCell model and a 2D CompuCell model. And here there's a system of 6 ODEs that will model the intercellular dynamics of the various signaling molecules that will force the differentiation. And here, the simple linear model just shows naive CD8 T cells coming in contact with APC. And in this stage, they will be pre-activated. And once the IL-2 receptor becomes activated, they will become activated cells. And then this TCR signaling right here will cause the production of TBET. And with enough TBET levels, the cell will become an effector cell. And this model, since it focuses on the early immune system, it does not include any memory cells or anything beyond 4 to 5 days. And my plan to go forward from here is to replicate the paper into CompuCell based on cellular and contacts between cells. And at the end, I'd like to put this project on NanoHub.
Okay, great. Thank you. Devin?
Yeah. So right now I'm still planning on working with the cancer stratification paper that we talked about. And so I have started to implement that in CompuCell. It's got most of the code in the paper that we worked with, working on kind of grinding that out and getting that working. And so once that's fully implemented, I want to work on expanding it. A couple of things that I've thought of. The current model only uses glucose as a chemical field to work with. So I want to think about maybe implementing like oxygen, pH, and other growth factors and stuff like that. And then also, like we talked about, tagging and observing mutations so we can better understand it and hopefully attack it with treatment. And then ultimately, once all that's done, just implement it on NanoHub. And that's all.
Okay, great. Jeffrey?
Okay. So first, we've been working on Pussyflow. I keep saying it, so this is a plot from the simulation and these are the parameters for the forces in the dissipative particle dynamics. And then I was talking to Andy yesterday, and he so those pictures were done not along with the simulation, so he asked me to pair it up. And so this afternoon I was working on that, and I didn't have a chance to make anything other than a screen share video. So this is the video of that we have of the plots being adjusted as the flow progresses, and it takes a while, so that's why I didn't just load the simulation now. But yeah, you can see here, this is what we have so far, and so we're going to start doing some more complicated things in this week. I have a list of my tasks to do, but I don't remember what they are right now. They're in a Google Doc, so I can't say, but yeah, so this is what we've done.
Okay, great. That's perfect. Drew.
Right. So I met with Joshua to go over my project on drug delivery for tumors, and we went over like the different what exactly I need to do for it to run, and I've been going over the paper to better understand the biology that's going on and some of the equations, and I think at this point it's just a matter of getting better at using CompuCell for me, because some of the equations are sort of complicated right now, but you know, we'll get help with that.
Okay, any other discussion right at the moment? No? Okay. So, I have to find the screen share. Here we go. Thank you. So I wanted to talk a little bit about what I was trying to do in terms of helping people structure their projects for the next few weeks. And I created something that I called Homework 5A. And did everybody get a copy? Can people see it? Is it screen shared?
Yeah, we can see it.
Okay. So I wanted to remind you where we are in terms of the semester. And ideally, people will have a first version of their projects ready on the 13th of April. And the reason we need them on the 13th of April is that gives us time to have one round of revisions and corrections before the end of the semester. And so I tried to, people have been working on these already, but I tried to give a little bit of a more formal outline of what I'd like you to do in terms of a statement. Now, in the case of Fanbo, that may not, the structure I have here is more for building a model of a specific biological system. And so some of this may not apply to the things that you're doing so much, but you could follow the general outline even so. And Jeffrey, also, you're not modeling so much as living tissue as a fluid, but some of the same issues will come up. And so I think the key things are to as soon as possible, try to have a specific set of biological problem that you're going to be addressing. And if possible, if you're working off of a specific paper, try to list the specific hypotheses that the paper evaluated, and what the main conclusions were and how they decided. In other words, if the paper tested a hypothesis, how was the decision about whether that hypothesis was true or false made? And then we'll come back to some of these issues in a minute. I'll talk a little bit more, but what are the inputs and outputs of the model? And if it's a paper that's validated against experiment, what are the experimental data sets that were used for validation? And then I'd like you to try to start building what I'd call a conceptual model. And again, we'll talk about this in a little bit more detail. What are the things you're modeling, the objects? What are their processes? What do they do? And then try to start building a table, which you'll refine with time, which gives a structured description of what the model consists of and what the interactions are. And ideally, that also should exist as the form of a diagram. And you don't have to be too concerned with the specific standards for annotation of the diagrams, but some diagram that looks like the things you would see in a biology textbook that explains how the components interact and what they do. And ideally, you'd like to be able then to begin to think about how those components interact quantitatively as well as qualitatively. There was actually a very, very good talk yesterday in this NSF panel I was listening in on by somebody who was from the US Department of Agriculture, Las Cruces, who was working on equine diseases and building very detailed models of the spread of a particular horse disease. It's carried by a variety of insects, so she had to have models of the weather, about insect replication, about how horses are moved around by humans, how they interact socially. A very, very elaborate model with a lot of experimental validation. And she was using, I thought very nicely, this same language of conceptual model, identifying the objects, things in the model, the processes involved, and then how you come up with at least a qualitative idea of how those processes can be described. And I had an example thinking about dictyostelium, although the dictyostelium example I've written out in this case is not the one that Kali decided to do. It's a different stage of dictyostelium. But I hope that if you look at that, it may give you some ideas about how to frame these ideas. It's not a trivial thing to do, to build these kinds of models, because they're very complicated. And one thing that I would suggest very strongly is that if you have models that have many different submodels, that you try to build the components independently and validate them independently. So for example, in a model of dictyostelium discoideum aggregation, you have amoeba that are distributed in the environment. Some amoeba become spontaneous emitters of pulses of a chemical called cyclic AMP. The cyclic AMP diffuses in the environment. When other amoeba are exposed to it, they move towards the source of the chemical and they send out a pulse of cyclic AMP themselves, after which they become non-responsive to cyclic AMP for some period of time. So you have a model of what happens intracellularly at a chemical level in terms of receiving and responding to the cyclic AMP pulse that sets what's called the refractory period. You have another model, which is how the amoeba move in response to the chemical in their environment. They stick to each other, so you have a model of that. And you would like to build these models separately. So you would have a model of the intracellular signaling mechanism, and you'd test that in response to a fixed amount of cyclic AMP in the environment. You'd have a model of the motility of the cell, and you'd test that in response to a particular gradient of the chemical in the environment. You'd have a situation in which the cells were fixed in position and could secrete and respond to cyclic AMP, and you check that that behaved properly. And you could test in each one of these cases how the key output, how long it takes for the cell to respond again after it had signaled, how far it goes in response to the chemical, what the wavelength and frequency of the oscillations in the space are. These are all things that one can test separately and check their dependence on key model parameters. And so building a substructure like that allows you to build the components of the model and test them separately before you put it all together. And with very complicated models, that's critical, because if you try to do it all at once and it doesn't work, it's quite difficult to find out what went wrong. And so what I'm asking you to do for next week is to try to give me an overview of what you want to deliver. We have basically 1, 2, 3, 4, 5 weeks, intermediate weeks, until you have to have an initial version of your project. And so what I'm asking you to do now is first create a weekly schedule of what you're going to deliver. And then each week, starting this week for next week, I want you to create a little bit of a self-assignment, which says that between now and a week from now, I'm going to achieve this part of my model. It could be I'm going to build the conceptual model, I'm going to build the model of signaling, I'm going to build the model of motility of the cell. And each week, I want you to hand in as part of your homework what you did on your self-assignment for the previous week. Of course, that can change. Sometimes you get more done or less done, something works or doesn't work. And then the second homework problem is assigning yourself the problem for the next week. And so you'll be graded on that. And that way you have a bit of a discipline and a reward for thinking about the logistics of the project. And so, for example, for the dictee, the way I outlined it, the first week might be creating an antimony version of the molecular signaling in the dictee and showing that the refractory period is correct for the parameters you picked and how it depends on those parameters. The second week might be fixing the dictee cells in space, putting that model inside each cell, and making sure that the relaying of the signal is right. The third week might be having the cells move and checking that the velocity of the cells in response to the gradients is right, and so on. I hope that helps a little bit, and that makes some sense. And if people have suggestions for ways to refine that model, that assignment, that would be fine. But I think we need to have some kind of structure, because 5 weeks is very short for building models of this complexity. I want to talk a little bit about how to think about conceptual models. I really wish I had the slide deck from the person who spoke yesterday at this NSF meeting, because she did a really beautiful job of it, again, in the context of geographical spread of horse diseases, which seems very different. But in fact, structurally, the models are very much the same. The first thing to think about is what a model is. And a model is something that takes a model structure and a set of parameters and gives you some output that you can compare to the real world. And that seems very, very abstract. But if you're going to have a model that does something, it has to have those components. And ideally, those models are as simple as possible. And if you talk to people like Alan Perlson, he spends a great deal of his life trying to simplify models. In our work here, we're not going to spend as much effort on simplification, especially if you're pulling things in from an existing paper. Then to some extent, you have the model structure given to you, and maybe the parameter values as well. But it's important that you understand on your own what those things are doing. And there's some language that you might want to think about. So you can think this comes from Andy. So his language is slightly different from the one I would use, but it's still very helpful. So you have, again, this model structure, a template, and then you have a state of the model, which specifies all the variables and parameters at the beginning. And that creates an instance or a replica, an instance of the model, which is that model populated with particular parameters, particular initial conditions. And then when you run it, because these are stochastic models, even when you run the same model multiple times, you're going to get different answers. And so you have multiple replicas of each simulation and the outputs associated with it. These are dynamic models, and one of the things that we'll do today, and Josh and TJ are on the call so they can help out with this a little bit, is showing you how to make movies using CompuCell and saving simulation output, which is something that we haven't really talked about. I noticed in some of the homework, people are actually taking cell phone pictures of their screens to try to save pictures of what's happening. And CompuCell has a reasonable, although not tremendously sophisticated ability to save movies. And so you shouldn't have to take a photograph of your screen. That really, we shouldn't have to make, if you're having to do that, then something's wrong with our code. And so, yeah, go ahead.
I remember for like the very first homework assignment, I would try to take screenshots of the simulation, but then it would crash every time. So then I just took pictures on my phone so I didn't have to keep reopening it. But I haven't really had that problem since. So that could have just been maybe NanoHub was being weird that day.
Okay, well, please, by all means, if those kinds of things happen, let me and TJ know about it so we can try to fix it. Because I understand the frustration. There were a lot of people who had it in cell phone pictures, so maybe everyone was having the same problem. But I want to make sure that everybody's on the same page on that one. And so once you have some kind of simulation, the simulation will consist of a series of states, and very often we'll take a picture and that picture may be actually an image, or it might be the readout of parameters or variables in the simulation at a given time. We often call that a snapshot. So we've heard some of this language before. When you're talking about models, a model to be instantiable, to be executable, has to always have 4 elements in it. You have to have the things that you're modeling, the objects, and their associated properties and states. If you have cells, then you have to say, for example, where they are and what kind of cell they are. So those would be properties, might be the type of cell, the state, could be where it is. Is it alive or dead? Is it replicating or not replicating? They're associated with processes, and those processes are divisible in parts. The behaviors and interactions of the cells, for example, and those behaviors and interactions essentially are potential, things that could happen. And then the initial and boundary conditions, what the initial configuration, the state of every object is in the system, and boundary conditions, what happens at the edges of the system, both in time. You can think of an initial condition as a boundary condition in time. What happens at the physical edges of the simulation. A lot of times we'll use periodic boundary conditions or no flux boundary conditions, but those are things one has to think about. And then given the object's behaviors and interactions in the initial and boundary conditions, we need something that evolves the system forward in time. Dynamics are the things that evolve the system forward in time continuously, and events are things that happen at discrete times. So dynamics might be the movement of a cell. An event would be a cell division because there's a discrete change between having one cell and having 2 cells in the system. And so again, when we think about this terminology, objects are the things being modeled. Typically in our world, there'll be cells, There might be subcellular component. There might be chemical species inside of cells. There often will be chemical species diffusing outside of cells. If we're doing Jeffrey's model, there could be particles of fluid that are moving in a pipe. So there are many different kinds of objects one could have in a simulation. The properties are the intrinsic characteristics of the object. Its volume, its position, its orientation, its mass. In general, we'll have a distinction between things that we call variables, which are results of the simulation, and properties, which are essentially fixed object characteristics. And together, objects and properties and variables describe the state of an object. And we can, I'm going to skip through this because to go through this in detail takes a lot of time, but I'll make the slide deck available if people want to go dive a little bit further. Since our objects mostly will exist in physical space, we have to decide whether they're discrete or continuous. Cells are discrete objects, chemical fields are continuous objects. But there also are non-spatial objects. For example, the chemical concentration carried inside of a cell doesn't exist in space at any given point. It's an abstract object. When we talk about behaviors and interactions, processes, there's also a little bit of an arbitrary division between the term behavior and interaction. A behavior typically is something that cells do, or the objects do autonomously. If I only had one object in my universe, my modeled universe, and by the way, the universe that my models live in is an object in itself. If I only had one object in my universe, one cell, what would it do? Well, the cell would probably have a volume regardless of what's going on around it. It might be able to move on its own, might be able to divide on its own. But there also are interactions, by definition, have to have at least two objects participating in them. So if I want cells to stick to each other, I have to have two objects because the cell can't stick to itself. If I want a cell to absorb a chemical from the outside, I have to have two objects, the cell and the chemical field. And so these behaviors and interactions are generally grouped into the concept of processes. And then in each case, what we're going to do, the objects of the processes together describe the core of any model that we're going to build. And so you have to think, and unfortunately, we don't really have good software tools for diagramming or describing these. So we tend to build tables. And so you have a table where the first column lists all of the objects of the system. And the second column lists all of their attributes, that is, their states, parameters, and variables. And the third column would list all of the processes that they participate in. And it's not very elegant and it's somewhat redundant, but those tables are extremely valuable when you come to debugging models later on. So again, if we think about and there's always some arbitrariness in how you make these definitions. So for example, if I wanted to model, I don't have a spring, I guess I can use this paperclip. If I imagine that I have two masses connected by a spring, I could either have three objects, one of which is mass 1, one of which is spring, and one of which is mass 2. Or I could have mass 1 and mass 2 and an interaction potential between them that describes the effect of the spring. And so in one case, I've chosen to represent the force between the two objects explicitly with a physical object of a spring. In the other case, I've represented that spring implicitly as a potential energy. And so those are decisions that you have to make in your simulation. And it's important that those things be written down and the justification for why you're doing them be written down. Again, for initial conditions, where things are, where my objects are, and what their states are. And again, remembering that the universe has a state as an object and has a state. And so things, global variables like the size of the cell lattice or the temperature, the fluctuation amplitude in the simulation, those are attributes of the simulation as a whole. And then again, the boundary conditions, what happens at the edge of my simulation, because I'm not simulating the whole universe, I'm only simulating a box. It's a piece of it. In our world, dynamics will typically be given by, for cell objects, will be given by that Potts metropolis algorithm that we use. If we have other kinds of object classes like chemical fields, then we'll have diffusion equations that govern their dynamics. We might have subcellular models that are chemical reactions, and they'll be governed by ordinary differential equation kinetic models, or maybe stochastic ones. And those kind of dynamics are things that we're pretty used to and we take for granted a bit, but which Andy's code is thinking about in a lot more detail. And then we have what are called events, which are discrete changes in time when something happens abruptly in time. We add a chemical to the environment at time X. We kill half of our cells at time Y. A cell divides at time Z. Anything that happens discontinuously in time is an event. And then the state of the whole model is a result of those initial conditions and the states of the objects and their processes and the dynamics. So the state of the model at any time after the initial condition is not part of the model specification, it's the result. And when we're going to build models, also in this class, we've taken some shortcuts. And it's a little bit like it used to be with airlines, where the airline would say, well, we make you board at a particular sequence because it's more efficient. Remembering the days when people actually got on an airplane. And then sometimes they would say, well, we're in a big hurry, so we're going to have everybody get on at the same time. Those can't both be true. In other words, if it's more efficient to board in a particular order, then if you're in a hurry, you should follow that order. If it's more efficient to let everybody scramble, then in principle, you should do that. At least the justification isn't right. And so in terms of model development, we're in a hurry here. And so we've taken some shortcuts, but in fact, those shortcuts may slow you down in the long run. So when we're going to build a model, we start out with some set of biological observations. Those might be things that you make or that you read in the literature, or if you are starting from somebody else's paper, they're observations that were made by somebody else. And then you have to define some initial set of questions and hypotheses. And a question would be a broader thing than a hypothesis. Hypothesis might be that P53, if I knock out P53 gene, the cancer will grow faster. That would be a hypothesis. The question would be, why are some cancers faster growing than others? The hypothesis has to be framed in the form of a yes or no question that can be answered definitively. A question could be something a bit broader in terms of understanding. Hypothesis can't have the word understand in it. Hypothesis is a yes or no question. Then we're going to define what I call a qualitative verbal model, or otherwise known as conceptual model, which is the thing that biologists love, and biologists like to encapsulate them primarily in the form of diagrams, pictures, but they can also be in the form of tables. And the table of objects and processes that I want you to build is a qualitative verbal model. Then you have to define how that model translates into something mathematical. And ideally, that mathematics is something that isn't tied specifically to the CompuCell instantiation. For example, that the idea that cells move in a particular direction with a particular velocity is not tied to CompuCell. That's an externally quantitative measurement that you could make on the real cells as well as on the simulation. You build then a set of method-independent parameters. For example, given a particular gradient of a particular chemical measured in nanomolars per micron, how fast does the cell move in microns per minute? That would be a parameter. And that's not tied again to the particular model specification, the way we build the model. And then once you've done that, you have to think about how to translate that into the particular things that you have to do inside of CompuCell and implement it as a simulation. And so the key thing to begin with is to think about what the questions are you're trying to answer. And when you're reading those, ideally you start defining the template, which is what are your objects, what are your processes, what do you think is important in the system? And drawing lots of pictures is a good way of trying to process that information and documenting it. Then we need to be able to define the questions we're going to ask and the hypotheses. And I think one of the things that is hardest to do, and I see this on the output side, is when I ask a question in the homework about what do you see when something, you do two simulations, the question is what do you see? And those kinds of questions, which are quite challenging, are designed to help you think about this question of definition of hypothesis and hypothesis testing. And so I'm not going to go into all of the details here, but I'd like you to come back to this slide deck. And there is a printed version of this as well, which was distributed, I think, at the very beginning of class on how to build models. Once you've defined what your questions are, what your hypotheses are, hypothesis could be something like a mutation in P53 gene causes the tumor to become larger. That's a hypothesis, testable hypothesis with a yes or no answer. If I have that hypothesis, I have to have cells that are growing to make the tumor, and those cells have to have some P53 representation of what P53 does inside of them. So the moment you specify a hypothesis to test, you've said a lot about what the model structure has to be, what objects and processes you have to include, what parameters you have to include, and also what things you have to measure in the simulation to be able to validate, to say whether that hypothesis is true or not, whether it agrees with observation or not. And so the conceptual model then is an embodiment of all these questions and ideas that you have. Once you've done that, again, you try to translate it into mathematics. For example, if you have a chemical reaction, the fact that you have a chemical reaction is a conceptual model. The fact that a chemical reaction occurs according to mass action or Michaelis-Menten kinetics, those are specific quantitative model structures. They're particular impositions on top of the qualitative model. Once I have something like a rate law, then that rate law is associated with parameters. If I have mass action kinetics for a chemical reaction, I have a single parameter, which is the rate. If I have Michaelis-Menten kinetics for a reaction, I have a rate and a Michaelis constant. And so the particular form of mathematical relationship that I've defined will tell me what parameters I need. Something that's quite important, and I got into a little bit of a headbutt with somebody today in this NSF meeting about, is that models should be shareable. People should be able to understand them and reproduce them. That's part of what point of the exercise that you're going through in this class. And the kind of modeling that we're talking about, the qualitative model is not tied to a particular methodology. The qualitative model should be able to be translated into CompuCell, into PhysiCell, into Morpheus, or into any multi-scale modeling environment. The method-independent parameters should also be independent of the modeling methodology. And so anything that only exists in CompuCell, for example, the lattice size, which doesn't exist in PhysiCell, that's a methodology-dependent parameter. But the volume of the cell has an objective reality that's not tied to being in PhysiCell or CompuCell or Mechanica. And so these should always be things that everybody who looks at the system can agree on. It's not associated with the computational way you're solving the system. Then when I do the simulation, I have to say, okay, now I'm going to implement my cells in CompuCell or Mechanica or PhysiCell. And now I have to make decisions about the particular ways the computing is done to represent those things, to represent those objects and those processes. And then things aren't shareable anymore. And now we're going from the world of biological and mathematical description to the world of computing instantiation. And then, of course, we have to actually build the simulation. And that simulation, in the case of CompuCell, is always going to be stochastic. In the case of Andy's Mechanica, it could be stochastic or it could not be stochastic because you have direct control over the fluctuations. I believe in PhysiCell, you have the same level of control. And then we have to ask the question, how do we use the outputs of our simulation? What are the metrics? What are the things we measure about the simulation that allow us to determine whether it agreed with reality or not? And that's probably the hardest thing. And that should have been done at the very beginning. I say, how are we going to determine from the output of our simulation whether the simulation replicates reality or not? That's the hardest thing to do. And so if I looked at cell sorting, we saw before we have two kinds of cells. They stick to each other, they move randomly, and they sort out. And so if I were going to ask the question, what are the objects in this? Well, I have two kinds of cell object, black cells and white cells here. I have some extracellular environment, which is medium. It tells me what my objects are. And the question I'm trying to answer is, can differential adhesion, the degree to which cells stick to each other, which is dependent on the type of cell I have and the amplitude of fluctuations that I have in the system, is that sufficient to explain the kinetics of sorting out that I see in these experiments? And so again, in this, I have two cell types, dark and light here, medium. And then I have to say, what are the object properties? In this case, I have to just accept from experiment that the cells aren't growing or shrinking during the experiment. So they have fixed volumes and surface areas. They're not dying or being born during the experiment. I'm not adding or subtracting medium. And I don't see any internal structure, anisotropy, or anything else. So my cells are behaving like little balloons that seem to be uniform, that are fluctuating. And the medium doesn't seem to do anything. What I do see is that the cells stick to each other. I can see that they stick to each other because the cells are in a clump. They're not distributed in the medium. And the hypothesis is that the relative adhesion between dark cells is higher than between light cells. And they both repel medium. And that's why I get the patterns of behavior that I see. I also, if I looked in a little more detail under a microscope, I would see that the cells fluctuate and move around like a little amoeba, and that the medium is passive. And my initial condition was a random mixture of dark and light cells surrounded by medium in a clump. And so I asked about building a diagram or a table to describe this. And this is what a table might look like. This could be written out in a little bit more detail because I could describe it for each cell type independently. But here's a list of my objects: dark cells, light cells, and medium, and a list of the cell properties. The cells have fixed volumes and surface areas. They don't grow, shrink, divide, or die. The amount of medium is constant. The cells are isotropic. All of the dark cells seem equivalent to each other. All the light cells seem equivalent to each other. The cells stick to each other, but not so much that they can't rearrange. The dark cells stick to each other more than the light cells, and light cells stick to each other more than light to dark. Both type repel medium. Dynamics is cells undergo random amoeboid movement. There are no events. And my initial condition is a random mixture of cells in a sphere. And so that could be written out in the form of a table. And there's a table that would do that. My environment is the world in which I'm going to put my cells. In this case, I've made it 2-dimensional. My cells have to have a position. They might have a mass. They certainly have to have a volume, a surface area. Medium has some volume associated with it. The cells also have a type. Here I've called them endoderm and mesoderm. They could have called dark and light. And so this is the kind of tabular organization that you can use to build model description. If I want to drill down a little bit more, the cells have some kind of behavior called movement, which is random. The cells are volume excluding. They don't overlap each other. We get that for free once you have the volume plug-in in CompuCell, but we're not supposed to be thinking about that yet. The cells stick to each other with an adhesion strength per unit area. The cells stick to the extracellular environment with some adhesion strength per unit area. And so you can begin then to drill down and build a table that describes all of the ideas that you've embodied in your model. If we want to make this more quantitative, we could begin to think about the functional forms for adhesion, for motility, and so on. And so that really was as far as I wanted to go with walking you through that. I have more slides, but I hid them because I don't want to take the whole class on that. Any questions about that? Because this is, I understand that this kind of discussion seems pretty abstract to begin with, but over the years of building and running dozens to hundreds of different models, I really believe strongly that thinking about things in this way rather than jumping in and coding will help you build models that work and that answer the questions that you want to answer. And you'll find that some of the papers that you may be referencing will do things in this way, others may not. If they do things in this way, you will find that replicating them is easy. If the papers do not do things this way, you may or may not find that this is easy. A couple of years ago when Josh took this course, he picked a paper that seemed to be rather simple to replicate, and it took him 1.5 years to try to actually get the replication done. And the reason was that that paper jumped into computer code immediately and never really explained the decisions it was making about how its model universe worked and what it was trying to do. And so if you want the work you do now and in the future to be something that other people can understand, following this kind of framework in terms of presentation is really pretty important. Okay. So now I want to come back to a quick review of what we did the last time. It's 2 weeks ago, so it's a long time ago now. We had originally specified cell volumes and cell surface areas, and we did both of those things in terms of constraints in the CC3D-ML. But if we want to be able to manipulate them at a cell-by-cell level, we have to specify them in Python. And that means we still need the XML to specify the plugins, but now we don't specify the parameter values in the XML. We just say that the cells have a volume, plugin name equals volume is saying so, asserts the cells have a volume. plug in name equals surface, asserts the cells have a surface area. And then inside of our CompuCell steppables, we are going to set the parameters for each cell. In this case, the target volume and the inverse compressibility, lambda volume, the target surface, and the inverse elasticity of the surface of the cell, lambda surface. And one thing that we've talked about and that you've worked with a little bit and we'll use again today is that having defined things this way, the pressure inside a cell is -2 times lambda volume times the difference between the target and the actual volume. And pressure, whether you define the pressure as pointing outward or inward, is up to you. And so that minus sign is something that's sort of up to you. You have to decide which is more convenient. And there's a membrane tension or a surface tension, which has the same form, -2 lambda surface, the actual surface area minus the target. And then in the homework, you were asked to do a couple of exercises related to that. Something that we came up with that we talked about that was quite important and useful in CompuCell is adding attributes to cells. And we do that through a Python dictionary. So every cell has a dictionary by default. And so cell.dict, if I put square brackets and then the name of some attribute or variable that I want to attach to the cell, I can set it equal to something. And these attributes could be strings, they could be numbers, they could be lists, they could be dictionaries, they could be other complex objects. It's powerful, but you do have to remember what you've done. One of the things that we run into in CompuCell, and we're going to run into it today, is that information is not automatically shared between different steppables. And you could say, well, why do I need more than one steppable? You could, in principle, put your whole model description into a single steppable, and you wouldn't have this problem. But as you build more and more complex models, you typically want to have modules that you can use independently of each other. And so those tend to get wrapped as separate steppables. And CompuCell recently, quite recently, solved this problem of sharing variables by creating a dictionary for the whole simulation called shared_steppable_varies. And that probably should have been something like self.simulation.dict to follow the syntax cell.dict. But in any case, it's a place where I can create variables that are available between steppables. And that's something that you're going to find you're going to use quite a bit. And I may forget to use it because it didn't exist until fairly recent. And then the last thing I wanted to remind you of was tracking fields. Again, we're going to use tracking fields today. Once cells have a dictionary entry, if it's a scalar, so a numerical value, if I put into the init function self.track_level cell_level_scalar_attribute, field name, can have it anything I want, and then the attribute name here is the name of the dictionary entry, CompuCell will automatically display each cell colored in with a color that corresponds to the numerical value of that dictionary entry. So if I want to monitor the pressure in every cell in a simulation, if I give the cell an attribute, cell.dict, quote, pressure equals -2 cell.lambda volume times cell.volume minus cell.target volume, and then I set a tracking field, the system will automatically display for me the pressure inside every cell. And that's, I think, again, one of the most powerful things that CompuCell does. There are many things that we could wish it did more efficiently, but that one really works well. So now I'd like to talk just a little bit about the homework. And I was originally going to go and do some exercises together. And I'll leave it up to you to decide whether you want to take some time to do that or not. So in homework 3.2, the point of homework 3.2 was to explore surface and volume constraints. And I had asked you to first plot, create some cells, and then to plot the actual volume and surface area of those cells, all of those cells in the simulation. And this is a reproduction of one of the homeworks that was handed in. And then also average it and plot the average versus time. Now what you see here is a series of horizontal lines corresponding to, and there was some confusion about why you got this kind of line, these horizontal lines. The average, the green line here and the red line for the target surface and the target volume seem to be well behaved, but there was some confusion about why we got these apparent horizontal lines and what was going on. And what you have to do to understand that is to remember that each one of these, if I have, say, 200 cells, in the first place, the surface and the volume are both integers. And so they're going to be on discrete values. And the second place is if I have several hundred cells, there's going to be some range of possible volumes for the cell because there are fluctuations. And over that range, over most of the time, there are going to be at least one cell with every possible value. And if I have several hundred cells, in this way of plotting, I can't tell how many cells I have sitting at each one of these points. There could be 50 of them. And I'll notice that for the extreme values, it's probably hard to see in this picture, sometimes there's a cell with that value and sometimes there isn't. If I followed a single cell, what I'd see is its volume will jump up and down a little bit and fluctuate around the mean. And then I asked you to do some simple experiments where you start out with cells that are relatively equilibrated, where the volume and the surface area are about balanced with each other. And then I asked you to increase the surface area a lot. If you increase the surface area suddenly by a lot, the cells fall apart. They keep the same volume that they had before, but they create a lot of surface by falling into little droplets, each one of which is surrounded by medium. And so you get what we would call dust. The cells disperse, really more of an aerosol than a dust. In that case, the actual volume is close to the target volume. The actual surface area is close to the target surface area. Now, if I went the other way and made the target surface area very small compared to the volume, I basically now have something like a basketball where the gas or the material in the cell is very, very compressed and the cell becomes very, very rigid. And it will line up a little bit on the axes of the lattice, which is a lattice effect. In this case, the volume will be less than the target volume. This is a typo. The volume will be less than the target volume, and the surface area will be greater than the target surface area. It's correct here in the next line. That wasn't deleted. If I adjust lambda surface and lambda volume, the degree to which those mismatches occur change. If lambda volume is much bigger than lambda surface, then the actual volume will be closer to the target volume, and the actual surface will be further from the target surface. If I increase the target volume rapidly, so there isn't enough room for things, what I'll find is that the cells in the center of my cluster don't have any room to grow, and so their actual volume is smaller than the ones at the edge of the cluster. And that kind of compression does occur biologically. You'll also notice that if I look at this picture, the mean volume and surface are changing gradually with time. And that reflects the mechanical relaxation of the system. When I make a change, the system doesn't equilibrate instantly. It takes some time for it to equilibrate. And so I think in the interest, Josh, what do you think? Should we try to do this exercise or should we come back to it or skip it? I guess Josh stepped away from the computer for a bit. Okay. Okay. Well, I think we can come back to this. Do people want to walk through this exercise together, or do you think it's okay? Maybe we should just do it together. Do people have put it running quickly? So I'd suggest everybody fire up Twit very quickly. It'll take us about 10 minutes to do this exercise if we do it together. So why don't we try Twit. Let's go to Twit. And I'll create a new simulation. And we'll call it volume simulation, for example. So I'd like everybody to do this. And what I want you to do is we'll create a group of cells. We'll give them a target volume and target surface, and we'll see what happens when we change it. And in this one, we're not going to use the Python settings because we want to use the steering panel to adjust. So we'll just do volume simulation. We'll make it a 3D simulation. And we'll use blob initializer. Make the pixel copy range 3. I'll just have one cell type, which we can call cell. No chemical fields. And we'll use volume, surface. We don't need a contact energy. Although we can certainly set one if we want. And I'm trying to think if there's anything else we need to do. I think that's about it. Okay. And now in the XML, I said that I want the surface area to be 150. And the target volume to be 20. Sorry, I want let's see what I have here. I want to replicate what I have here. So I want to cell volume 125. So we'll say target volume is 125. Except here I said, here I said, we'll do steerings. Okay, so target volume is 125 and target surface is 150. And I'm going to use blob initializer to give me some cells to begin with. And we'll make the width 5, which is 5 by 5 by 5, which is what we want. Let's give them a little bit of space to be in, so make a gap of 2. And we'll run that. and we'll visualize it in 3D. Okay. And I'd like everybody to do that. Is it okay? So everybody please try that. Okay. It's running really slowly today. I guess NanoHub is not going to be very cooperative for doing larger simulations. So maybe I should have used a smaller simulation to begin. Given how slow that is, I'm going to, in the interest of time, I'm going to go back to our simulation. I'm going to make it half as big. I'm going to make it 128 cubed. It should in principle run 8 times faster. And then I have to change my blob initializer to put my blob at the center. If it's running fast enough for you, what you have is all right. Hope this runs a little bit faster. Yeah, that's better. Okay. So now, if you've got that working, you can open up the volume plugin and the surface plugin, and you can change the target volume from 125 to 25, and the target surface from 150 to 25. And you see that things fall apart at first, and then the cells recoalesce, and I have little tiny cells that are floating around. Are people able to see that? Did that work for people?
We can see it.
Okay, good. Now, I'd asked you in the homework to plot the mean volume and surface area. I think most people were okay with that, but let's just review quickly how to do that. To plot the values, we go to the steppable. Inside the start function, we would add CC3D Python, scientific plots, setup, and that will create a plot window. It happens to be called average volume and surface by default, so I don't have to change it. The only thing I might want to do is plot this, the mean volume by dots instead of lines, and change the size of my second dots to 5. Okay. And then inside the step function, I need to calculate my average volume. So I'm not going to do this. You can do this in a more Python-y, better way, but I'm not going to do it. Let's just call, we wanted mvol was the mean volume. So I'll set mean volume to 0. I'll set m surface to 0. And then for cell and self.cell list, I'm going to say mvol plus equals cell dot volume, msurf surface plus equals cell dot surface. That gets my total volume and surface. Outside of my for loop, I need to normalize by the number of cells. And so I say mvol divided by equals, and I can do float of length of self.cell list, which would be the number of cells. And the same thing for the surface. I'm always a little bit concerned with type changes in Python. You probably don't actually have to do that float. I'd rather do that. And now I need to add those 2 points to my plot. So again, I go to CCD Python scientific plot, add data point, and it's already set up. This one is actually plotting every data point. I only want the means. So I'll copy mvol here. And msurf. Yeah. Okay. Let's try that. And again, you should probably try doing the same thing. Oops. It doesn't like divide equals, all right. I thought divide equals worked. All right. What is the problem? Invalid syntax. Oh, I can't use the name that I gave for the plot. Let's call that MSIR1. Try that. All right, Josh, are you there? You see what I'm doing wrong? Oh, it means there's a parenthesis missing. Ah. So G has no attribute volume. Another typo. Let's see. Oh, typo. No. As I say, probably you can do these faster than I can. Not my day for typing. I hope people are doing this in parallel rather than watching me do it. There. Okay. So I hope that does everybody have. So people should try to do that. As I say, you could probably do it faster than I can, given the typos. So you're plotting mean volume and mean surface area versus time. And a couple of people in the homework had an issue with this. So if there are any questions about what you need to get to make it work, again, my typing was bad, so I didn't make it elegant. I didn't do it very elegantly. But. And the divide equals would have worked because the issue was my parentheses rather than that. Okay. So now, if you've got this, if you're still working on it, that's fine. Keep working on writing it. If you have it working already, you can now do this change that we made. I'll go from 125 to 25 for both of these. And I can see how the average volume and surface change when I make these changes. And the second one, I must be plotting something wrong here. Answer. plus equals on that surface. M01 should be okay. All1, all1, 01, all1, 01. Yeah. Okay. So now I could play around where here I have the target surface is 25. The target volume is, actually, why is that not? Something isn't right. I've got the average volume correct, but the average surface is not working. Let's see what happens if I change the average surface to 125. The red one is the average surface area. Red one. Oh, I changed the volume there. Okay. So that red one is volume. But the green one should be the average surface. It doesn't look like it is. So I've miscalculated. I've miscalculated the surface area. Let's see why. M sur 1 equals 0 plus equals cell dot surface. M sur 1 equals M sur 1 over float length cell list. That should be right. Adding M sur 1 to the list. So the volume is red and the surface is green. And so somehow I'm not plotting the surface area properly. Did other people get that to work? Because if you did, I'm not going to take time to keep you waiting for me. The average volume is working correctly. The average surface is not plotting. I wonder what I did that made it not add up. Let me try again.
It might be because you have equals plus instead of plus equals.
Oh, okay. I told you my typing is bad today. Thank you very much. I'm surprised equals plus doesn't throw an error. Why? What shouldn't it? Shouldn't that throw an error? Thank you. All right. Now something is. What did I do wrong now? Plus equals. Oh, because I didn't. Answer. I'm really not doing okay. Yeah. For you, 10 minutes. For me, half an hour. We'll get it eventually. There we go. All right. So now again, I can come in and I can change the target volume and surface. Let's make them 25. And now it's working. And what you see is it takes some amount of time for it to equilibrate. When I make these changes, they happen, but it takes, look at this gradual relaxation as the system is reorganized. You'll also see that target volume is 25 and target surface is 25, but the actual volume and surface are not quite right. The green was surface area. Surface area is a little bit bigger than 25. The actual volume is smaller than 25. If I make the target volume now 125, the cells are going to be very compressed. Notice the target surface is going up and the target volume is going up, but not enough. So the actual volume is much smaller than the target, and the actual surface is much bigger than the target. And notice that the cells have gotten very compressed and square. If I make the target volume something like 500, which is a bit ridiculous, you'll see that the average surface and volume both go up. The average surface has been pulled way above its target, but the average volume is now on the order of 180 when it should be 500. So the cells are very, very compressed. And so we could play with this a lot. We could put the target volume back down to 50. Things won't be so crazy. Let it settle down for a minute. And now let's make the surface area bigger. Make the target surface 125. Notice the surface adjusts almost instantaneously. The green line comes up. The volume comes up a little bit, but not much. And the cells become very broken up. If I make the target surface something really large, like 250, it adjusts right away. The volume doesn't change very much, and I get dust. So if the target surface is much bigger than the target volume, I get dust. If the target surface is much smaller than the target volume, I get rigid square cells. If the target surface is much bigger than the target volume, the target surface is approximately right. The target volume is approximately right. If I make the target surface small compared to the target volume, the volume is less than it should be, and the surface is more. And so this allows you to explore the degree to which things are mismatched and equilibrated. And you could also see the amount of time in terms of Monte Carlo steps that the system takes to reach equilibrium. In this case, it's taking about 10 or 15 Monte Carlo steps to equilibrate. Okay. So that was something that I wanted people to look at. And now if I want to save things, I go up to visualization. Sorry, not visualization. I go up here to the settings, configuration. And now I could say, save image for save visualization, tell it how frequently I want to do that in Monte Carlo steps, every 10 Monte Carlo steps. Hit OK. And then, if I remember correctly, this current version of CompuCell, Josh, you have to hit this button here, actually make it take the movie.
Right. You gotta hit on the camera button.
This camera button.
Make sure that it's safe.
And the reason for that is that it's got that you're telling it that you want to save images, but it will, if I change the view, may have crashed on me, but if I change the view of the image here, that what is rendered will change. And so the movie that I'm taking will be snapshots of what I'm seeing in this window. And if I want to configure it by changing the magnification, I want to change the angle of view, the color, and so on. I have to tell it what pictures I want. So here I've zoomed in too much. If I want to zoom out, I could do that, or if I want to take a 2D image, I could do that. If I want to say the average volume and surface, I'm not sure why it's hung up. I can also export. I could also do an export here, assuming that it catches up with me. I could do an export of the data set and save the data set as well. So I could save this plot as a CSV. Okay. Okay. So we also did differential adhesion, looked at 2 cell types, and that was the homework for this week. And we've talked about that at some length. I hope everybody was able to get simulations that look like this, exploring the effect of different cell-cell adhesion and the possible configurations. And even with just 2 kinds of cells in differential adhesion, you have rather a lot of possibilities in terms of configurations, from random mixtures to 1 cell type engulfing the others to what's called incomplete sorting or partial cell sorting, checkerboard, cells distributing themselves in the medium. And so those were all different configurations that we could get just playing with those adhesion. We also talked a little bit, and I don't want to spend a lot of time on it here, about how to measure cell sorting. In the case of cell volume and cell surface that we've been looking at here, it's relatively straightforward to calculate the mean volume and the mean surface area. If we want to count how much we're in contact with a cell of a given another type, it's a little bit more complicated. There is a function in CompuCell which returns the contact area of a cell with its neighbors given by cell type. And so we could do that. To do that, we have to do this function get cell neighbor data list, which is not so convenient. And there's also an iterator that would allow us to iterate over all the neighbors of a given cell. And then we could add things up by hand. And I don't want to spend a lot of time on that one here. If people had trouble with it, we can review that. So I'd like to now, let's take a 5-minute break. We're halfway through our class. Let's take a 5-minute break. And then when we come back at 6:45, we'll start on cell proliferation, which was really the main thing I wanted to cover in class today. Okay, everybody, ready to get going again?
Yep.
Thank you. Okay, so now we'll come back to cell proliferation and division. And actually having this here will probably be a little bit helpful. And I'm just remind you that we talked about cell proliferation. In reality, cell proliferation is determined by a lot of very complicated chemical factors. One thing that we're going to think about today in our exercises is contact inhibition of proliferation. So one of the key things that cells do is that when they're not in contact with each other, they divide to fill the space. And when they're touching each other, they send signals that say stop dividing. As a surrogate for that, we're going to use the pressure, which isn't a very good surrogate, but it's not a terrible one. In reality, also, cells have a complex cell cycle. We don't have a model of chromosomes and all of the complexity of going on inside of a cell. We're just going to have two things going on in our models. The cells are going to grow. And then while mitosis actually takes some time, and is sometimes incomplete, we're going to assume that cells divide instantaneously when they divide. And so the reality, again, of cell division is rather complex. We can see the beautiful dance of the chromosomes. But we also see that the time for the cell actually to split, so telophase is pretty short compared to the other cell cycle time. So assuming that it's instantaneous isn't that bad an assumption. Our basic idea is going to be that we're going to have the cell divide when it reaches a specified size. And so if we're going to do that in our simulation, we're going to have the cells grow. And to do that, we're going to have the volume specified in Python rather than in the XML. And so let's do that. This code is a little bit more complicated perhaps than it needs to be because it draws the cell by hand. We could use blob initializer instead. So what I'd like you to do is start with a single cell and have the cell volume grow in time. And we could do this in 2 dimensions. So this is a little in-class exercise where the assignment is to create a single cell and have the cell volume increase in time. And I would recommend to begin with that you define a contact energy with the medium, positive contact energy with the medium, and don't define the surface, cell surface. We're going to find out that if we have the surface, we have to adjust the surface to match the volume, and that makes things a little bit more work than we'd have to do this way. Is that clear what to do? And here's
I wanted to add just that yeah, be very mindful about commenting out the XML data for the volume, because otherwise anything that you do in the Python file cannot override the values that you assign in the XML.
So I was going to actually do this from scratch rather than use the one that I just had. Although actually having the maybe I should let. What do you think, Josh? Should I do? People should do it the way they prefer. You could do a new one, a new simulation, or you could use the simulation that we already had. I'll create a new simulation and then I'll also modify the one we have, and you can do it yourselves either way. So if I call a simulation cell growth, for example, I'll do it in 2D. As usual, I'll change the neighbor range. I'll use blob initializer. I'll have a single cell type, which I'll call cell. And then no chemical fields. I'm going to create a contact energy. I'll use volume local flex, which is the one where I specify the volume in Python. And I can actually create a growth steppable, which I may or may not want to do. uh, there is this cell growth steppable, which is on the screen here. So I'll actually hit growth steppable here like that. And is there anything else that I need? I don't think so. Okay. If I now look at my code here, cell growth steppable has already created the code to initialize the volume for me. So it iterates through the cell list. And it's also put in something where the cell target volume increases by 1 at each time step, which is very, very fast, as we'll find out. And the only thing that I need to do in my XML is if I want there to be a single cell, I need to go to blob initializer and make the radius of my blob small enough that I only get 1 cell. Okay. Let's see if this will redraw. Well, again, you may be able to do it. You may be able to do it faster than I could do it. So let me try launching it in Player again. Well, I can close it. Let's see if I can. Player seems to not want to cooperate. Let's see if I can. All right, open in Player. Ah, that's what happened. It staled my credentials. All right. I don't know why I did that. Okay. Yeah. Are people having trouble with NanoHub, or is it working for you?
It's working.
All right. I'm not sure what happened here for me. Player does not want to redraw. And if I hit terminal, I'll just start over. I'll do it. If you had something, if it was working for you, you should have gotten something that looks like this, where they start with one cell and the cell grows. Were people able to get that? Or did you get something else, or did it not work? Let's see if I can open that. I could open the other one. I don't think I saved the other simulation. But it was fast enough to generate. So growth. Okay. Okay, did everybody get that? Does anybody need help with that? Okay. So now the next thing we're going to do is implement a surface constraint. And if we implement a surface constraint, we're going to have an idea of what's reasonable. And so what I'd ask you to do is use the tracking field idea that we had before and keep track of the pressure. So add the pressure as a dictionary entry to the cell, and then have the pressure displayed as a tracking field. And I'll walk you through it, and then you can do it on your own. So the first thing we're going to do is we're going to create, when we create the cell, we're going to create a cell dictionary attribute, cell.dict. And the way I'm typing today, maybe you have to catch me. We'll call it pressure. And that's going to be equal to, and we're going to need to update this. So it really should be probably in the step function rather than in the start function. Here we've got for cell and cell.cell list. We'll calculate the pressure. And the pressure was equal to -2 times cell dot lambda volume times cell dot volume minus cell dot target volume. So that should give us the pressure. And then to display that, you go up to the init. We'll get there eventually. The init here. And we go to CC3D Python, extra fields automatic tracking, track scalar attribute. And we'll call the field name pressure. And the attribute name has to be exactly what that dictionary key was, which was pressure with a lowercase p. And let's see what happens now if we run it. See if it works. Oops. Cell G. Oh, I typed a capital V. What? Nope. Missing times. All right. So now, if I go under cell field, or I could actually create a second window by clicking here. And I now have the attribute pressure. And the color of this will tell me what the pressure is in this cell as it grows. Now, at the moment, that's not very interesting because there's nothing constraining it. And I'm actually not sure why it's blinking out on us like that. That's not something I'm used to. Should be okay. If I zoom in, the maximum value is 16. So the pressure inside this cell is about 16. So why doesn't everybody try to create a tracking field and evaluate the pressure? And you can see, let me slide this over a little bit so you can see the. Okay. All right. So if we want, so that was the tracking field to monitor the pressure. Let's not monitor the surface tension. That's too much work for the moment. We don't, and we're going to run out of time if we do too much. And now let's add a constant target surface. So to add the target surface, we have to go back to our XML. And we're going to have to add the plugin. And to do that, we have to go CC3DML plugin. And we want surface. And it looks like I put it, okay, fine. And I don't want the lambdas here. So I'll put surface in like that, which gives me the Python specification. And then in the growth steppable, I have to set my target for a surface. Like that. Okay. And now if I run it, start. Mismatch tag. Oh. I sure hope people's typing is better than mine. You'll notice now that the cell is very compressed. And if I look at the pressure, it's actually probably it's probably a little bit difficult to see it. And I'm not sure I can zoom that. If I see these tiny numbers here, which you probably can't see on your zoom, the pressure here is 476 at the moment. And if I keep running it, that pressure is going to keep going up and up and up as the target volume keeps trying to get higher, and it can't because the target surface area is holding it back. So if I wanted to, and let's, for the moment, let's skip adjusting the target surface to keep the surface tension constant. Adjusting the target surface to keep the pressure constant is something we could do. But let's, for the moment, let's just let the target surface, the surface constraint be 0. And in that case, the cell will grow freely. We can adjust the target surface later if we want it, but by setting lambda surface to 0, there's no effect. So let's see what happens if we do that. And again, this number is probably too small for you to see, but it's 10, 12, 6, 8, and so on. So the cell is now growing because there's no surface constraint. The pressure is small. Okay. So now what we are to do is think about cell division. And we have to think about how they're both biological questions about cell division and also CompuCell questions about cell division. So if we're thinking, the first thing about CompuCell cell division is that CompuCell specifies cell division using a special steppable we call mitosis steppable. And so while most of the steppables that we'll use are optional, if you want cells to divide, you have to have something called mitosis steppable. And so to do that, we're going to have to learn how to add a steppable to a simulation. Although if we use Wizard, it would generate it automatically. And then we're going to have to decide what is going to trigger cell division. And to begin with, we're going to assume that the cell divides when it gets to twice some target volume. In reality, there could be all sorts of complicated rules about cell division. And we're going to then make a list of all the cells we want to divide at a given time. And then we're going to go through that list and make the divisions. And the reason we're going to do that in 2 steps is that we have this Python rule that you can't, when you're iterating over a list, you're not allowed to change the elements of the list. And if we're iterating over all cells and we're creating cells, we're changing the list of cells, and that can cause that iterator to have problems. And so when we do cell division, we're almost always going to create a list of cells to divide, that iterate over that list to do the division. And so we're first going to iterate over all cells, check for a condition, stack the cells we're going to divide, and then divide them. We're not going to do it instantaneously. When a cell divides, we're creating an extra cell. We had one cell, we're creating two. And that means the first thing that we have to do is allocate the volume that belonged to the original cell to the two new cells. Or in this case, one cell will be the continuation of the previous cell, the other cell will be the new cell. And that will denote the cell that's continuing as the parent cell, and the new cell is the daughter cell. And so you have to decide which voxels of the original cell get allocated to which of the two. And CompuCell allows you to do that in a variety of ways. You could have it always divide along a fixed plane relative to the X, Y, and Z axes. That would be called oriented cell division. You could define any plane you like. You could have it divide in a random direction. You could divide along the long axis of the cell or the short axis of the cell. And so those are all pre-existing ideas. And then you have to decide that the parent cell continues, so it has whatever attributes it had before, state it had before. The daughter cell doesn't have any attributes by default. And so you have to decide what kind of cell it is, how big it is, and any other properties that it has. In particular, the target volume and surface area have to be defined. In general, if you think that the division is symmetrical, you'd expect that the target's volumes for the two offspring would be half of the original target volume. The target surface area with that represented membrane would be half of what you had before. But you could have asymmetric cell division as well. And then I remind you again that you may want to use that shared separable variables to pass information between steppables if you need to do that. So CompuCell has this function. If we did this in the wizard, there's a tick box for mitosis. We already used the tick box for growth, and mitosis will create the mitosis steppable for us. Now, when we do that, we've already seen how to do this. To do cell growth and specify the volume of the cell growth, we'll see there'll be in the Python, we'll see something called class mitosis steppable, and then there'll be the step function that we're used to. We'll create a list called cells to divide, square brackets. Here, we're going over all cells. We're saying if the volume of the cell is bigger than 50, we add it to the list to divide. That 50 could be some other condition. If cell internal value is bigger than 99, add to the list. If the cell gets too close to something else, add it to the list. We could add to have many different possible conditions. But this one is a simple one. And then we're going to go through the list of cells to divide. And we're going to then make the cell divide. We have a variety of options, as I mentioned. Random orientation, cell divided along a particular direction, cell divided along major axis, minor axis. And then this is the thing that's the most important, and is a very useful function, which is there's a function called update attributes. And update attributes allows us to specify for the parent cell and the child cell what properties we want them to have. Self.parent-cell.target volume divided by 2 says we're assigning half of our voxels to the parent cell. Self.clone parent to child copies every property of the parent cell over to the child cell. So in this case, the state, except for the position of the daughter cell, will be identical to the parent. If you try running this without attaching any attributes to the daughter cell, you'll get a mess because it won't have a target volume, it won't have a surface area, it'll just disappear on you. But this clone is very useful. In early versions of CompuCell, if you added things like dictionaries by yourself, they didn't clone. But now it copies over automatically. And that makes life really easy. So actually doing cell division now is much simpler than it used to be. We could also do this by adding a steppable in the editor. And now that I think about it, that's probably not present, is it, in the twinit we'll find out. So we're going to go to our Python, and then if we right click, there's add steppable. Good, it does work. So we select add steppable. And we want a mitosis steppable. And we can call this mitosis. And we don't need any of these fancy things at all. I'll just hit OK. And we will see, let's see if it put it in the right place. It did. So it's now created a new class called mitosis steppable. It's created a list, cells to divide for cell and self.cell list. If volume greater than 50, cells to divide append cell. For cells in cell to divide, self.random orientation, that's fine. Everything here is fine, except at the bottom of this update attributes, as a demonstration of how to make changes, it gives an example which says if self.parent cell type is 1, make the daughter cell of cell type 2. And since we only have one cell type in our simulation, that code would cause the simulation to crash. And so we're going to comment it out. Okay. And so let me save this. And so what should happen now if I run this? What should happen? Somebody? Let's try it. My cell grows, and when it reaches a volume of 50, it divides by 2. So I'd like everybody now to try running that simulation. So I realize I went through a bunch of steps, but the key thing is, does everybody have the cell growing, the growing cell? Does anybody not have the growing cell? If you don't have a growing cell, raise your hand or say something. Okay. So to now have the cell divide, you put your cursor somewhere inside of your existing steppable. You select the Python cell growth steppable here. You right click on it. You hit add steppable, and I don't want to do that a second time. Add steppable. Then when it pops up the steppable window, you select mitosis and hit OK. and it will paste in the template. When you look at that template, the one thing you have to do is at the end of it, it's changing the cell type from 1 to 2. We don't have a second cell type, and so you have to comment that out. And then you run it, and you should be able to see the growing cells. So I'll let everybody try that and see whether that works for people. If you've got any questions about that, please ask. So the template code was pretty good. The only thing that we had was the cell division was an issue. Again, if you want to do this, you go to cell growth steppable, right click, add steppable. So type a name for the steppable, click mitosis and OK. And then come down here and comment out these couple of lines at the end. And if you've got that already running, you might want to turn on the pressure field. And actually, pressure field is not doing what I expect. This was something else that happened in the homework, and I fell victim to it too. If I look at my color map for my pressure field, if I look down here at the bottom, it's too small for you to see. The maximum pressure is 450. If I look at my color map, my color bar goes from 0 to 1. And so I'm going to have to change the scaling of the field for the pressure field. And instead of having the maximum value be fixed, I have to make it floating. And now, when I run this, I can see the pressure. And what you'll see is the pressure is small at the periphery. And in the middle, these cells are growing. And remember, the cells are growing at a fixed rate. So I have exponential growth. And so those cells in the center are very compressed. And so the cells in the middle that are at very high pressure, the cells at the surface are at low pressure. That actually happens in tumors. You have pressure gradients like that. So I'd like everybody to see if you could get this pressure to work. And so you could see the pressure gradient in the tissue. So try visualizing the pressure and see if you get something that looks like that. I'll run it again. Again, the trick to get that to work is that you're going to have to make sure that the pressure is, that the field value for the pressure is auto-scaled. And to do that, you go to Settings, select Field, Settings, Field, and then make sure that the range is float, not set to be fixed. If people are having trouble with the tracking field for pressure, say something. Help them. If the cell is under pressure, it means its actual volume is less than its target volume. That means that if you were to change the doubling condition from cell dot volume greater than 50 to cell dot target volume greater than 50, you'd get a different result. So for the people who've got it already, try changing cell dot volume greater than 50 to cell dot target volume greater than 50. Try that and see what happens. It'll be different. See if you could think about how it's different. I haven't changed it yet. Let me try it with using the target volume rather than the actual volume for the division. Note if I divide on the target volume rather than the actual volume, the pressure is more uniform. Why might that be? The target volume increases in lockstep. So that means that the cell division happens in lockstep. The actual volume depends on the pressure. And so in one case, the system, the division is slave to the clock. In the other case, the division is an emergent property of the system, and there can be mechanical instability. And so you get actually quite a different result in those two cases. The other thing, if you're working on this and still, there's two people who still are working on it, I guess, you might try is what happens if you try one of these other cell division attributes? What if I do oriented cell division? Let's try that one. That doesn't seem to have made very much difference. I'm not sure why this pause button has disappeared on my screen. That's sort of odd. Huh. I'm actually not sure why I don't have a pause button. Never seen that happen before. Have you seen that, Josh? At the pause
I haven't seen it. I haven't seen it before, but I'm not very. To be honest, I'm not very familiar with the nano hub deployment.
Yeah. That's a new one for me. But in any case, you could play with the division options if you like, and see what you can do. Let's try again. So in this case, that's really odd. I don't know why. Why it won't let me? All right. It's going to be a bit dull. So here I've told it to divide the cells along a particular orientation. You notice it's dividing them along their, so that in the parallel to their long axis. And so that's one of the possibilities. You see they're dividing parallel to their long axis. In the end, it's not going to make a huge difference to the configuration. It's still pretty round. But when they divide, you can see that they divide in a particular way. If the cells were less motile, then you would see that more. Okay. So I'll put it back to random division. And I'll put the criterion for division to be volume rather than target volume. Okay. Now, if we look at the pressure and I let this continue to run, and this time I'm going to let it run and run. Might have been more useful if I actually had the plotted the mean pressure, the average pressure. What I'll see is that I have this high pressure region in the center. And when the cells get to the boundary and start bumping up against it, because this is dividing, the target volume keeps going up. As I run out of space, the pressure will keep going up and up and up without limit. And you see I'm getting all sorts of interesting mechanical instabilities in this, which are not biologically meaningful. They're rather interesting mechanically. If you have soap foams, you get this, but this is not how biological systems work. You see the maximum pressure keeps going up, 759, 7, and so on. Because I keep trying to put in more cells and there's no room. So in reality, most cells, most of the time, are not in cell cycle. Even in tumors, not all cells are in cell cycle at the same time. And as I mentioned, one of the most common ways that cells become quiescent out of cell cycle is due to pressure signaling. So let's do the first thing to do is let's create a second cell type, which we'll call quiescent. And now we'll use that switch that we had of the cell type so that one of the child cells of the dividing cell is quiescent. So why don't we try that together? We're going to have to go into the XML. And when we have our cell types, we're going to have to create a new cell type, which we're going to call quiescent. We can do that by hand. Call it cell type 2. And we'll call this quiescent. And we have to then, for contact energy, we're going to have to replace this contact energy. And to do that, we go to CC3DML, plugins, contact. And we can just do replace. And that will automatically paste in all the values for the new cell type. Okay. And then we go back to our cell growth steppable. And where we did this comment out here, we'll do block on comment. And then here I was very literal and we did the cell type by number, but we should really do it. We should really do it by cell attribute. And if we don't remember that, cell attribute is cell type here would be cell dot type. Let's see. At the rate I'm going to get things wrong again. So we want to set the cell type here. Let's see. If the parent cell was self dot, we called it self dot cell. We want the daughter cell to be type self dot quiescent. And we don't need anything else. And we only want the cell type cells of type cell to grow. So here, instead of for self and cell dot list, we're going to use the iterator for cells by type here. And so here we need this line, cell list by type. And so this will be just type cell. And the thing we have to remember is that the cell type name has to go in all. So is it clear what the exercise is? So you're going to create a new cell type with your XML. And I called that cell type quiescent. When you create a new cell type, you have to update the contact energies, although if you don't do it, nothing dramatic will happen. Then when you go into the cell growth steppable, we have to change only the cell type is going to grow. So we have to change our iterator here. And then the daughter cell, if it was a parent was of type cell, the daughter cell is of type quiescent. And so let's see what happens when we do that. Probably I'll have a typo. So we'll see. Yeah. There's no attribute type name 1. Right?
It seems like in the iterator yeah, right, in line 56 you need to specify.
But oh, for the volume also, right? Yeah,
Right.
I need the same thing for the volume. It was in the growth steppable. I don't want the quiescent cells to grow, although it's interesting to see what happens when they do. So it was, growth was here in line 30. And for this one, there's an issue, which is I didn't create a cell. This is a little more complicated than I want it to be. I have to now separate in line 30. The cell list, I want the pressure to be true in every cell, but I want the growth to only happen in the cells of type cell. So I have to split that. Again, you could do this your own way. There's no reason you have to follow what I'm doing. All right. So now the yellow cell is my cell of type cell. It's growing and dividing. It stays yellow, but one of the daughters becomes quiescent and just sits there. So why don't people try implementing that simulation? And if I look at the pressure field, I see that the pressure is more or less random. I don't have that much cell division going on. Fluctuates a bit, but not a lot. So I've gone from exponential growth to linear growth. So is it clear what you should try to do? You should try to, and again, I realize that this is a lot to do at once, but once you've done this once or twice, I hope it becomes easier. And I'd like to do it together so that if there are questions about it, or people have difficulty with it, we can talk about how to do it. So again, the steps to do this are create the quiescent cell type, which is done in the XML. And then in the cell division steppable, when our initial cell divides, its daughter cell, the child cell, becomes quiescent. And you could just do those two steps to begin with, and not change who's dividing and who's growing. That would be all right. And then once you've gotten that going, what you'll get is the salt and pepper color. So if I hadn't changed my iterators, what I would get is this. All the cells are dividing, but that yellow cell, when it divides, creates green cells. If I change my iterators back, I'm now only the yellow cell is growing, and only the yellow cell is dividing. Okay. If I don't change the cell types, I'm back where I was with the new iterators don't do anything. Okay. So I see a couple of people don't have this. If you need help, please speak up and we'll help you get through it. Because you'll definitely need this for homeworks. So, in fact, you'll need this, I think, for every problem, every CompuCell project will need this. That's been discussed. So it's important that we walk through it. And I realize that I'm covering a lot of material relatively fast. Okay. So we saw what happened here. And now we could ask the question, if we have this unlimited cell growth, we get this high pressure region in the middle. In reality, as I mentioned, the cell growth goes down as the pressure goes up. Contact inhibition. In fact, real pressure also inhibits cell growth. They're chemical signals, but they're also direct pressure signals. And so one of the things we could do is in our initial simulation, we had this very naive growth rule, which was that cells grew at a constant rate. But we could have that growth rate go down as the pressure goes up. And we could think about different rules that we could use based on the pressure to say how fast the cell grows. Clearly, as the pressure gets bigger, we want the cell growth rate to get smaller. The simplest thing would be something where the growth rate decreased linearly as the pressure went up. The only thing that's a little funny about doing that is that if we make that happen, then the growth rate will go negative if the pressure gets to be too big. But it's not an unreasonable thing to do. The other thing we saw was that the pressures were getting up to be values of 20, 40, 60. And so just subtracting the growth rate pressure from the growth rate would be too big. So we have to rescale it. So why don't you, as the last thing we're going to do, look at the pressure. Here the pressures are, let's start out early because when we change this, the pressures are not going to be so big. And let's look at a typical pressure in the simulation. Okay. Well, the pressure is already up to 200. So let's say that we're going to have the cell stop growing when the pressure is 40. So let us now, our last little project for this today is to replace cell dot target volume plus equals 1 with something where the growth rate goes from 1 when the pressure is small to 0 when the pressure is 40. And there are a variety of functional forms you could use, but you pick one. I'll do one on my own, and we can see how it compares. Let's see. I think that should do it. Let's check. If the cell, if this pressure in the cell is 0, cell dot target volume plus equals max of 0, comma 1, comma minus cell dict, that'll be 1.0, will be 1. If the pressure is 40, then I have 1 minus cell dict of 40 divided by 40 is 0. I don't want this to go negative, so I want to max it with 0. And so in this case, I should wind up with a situation in which the cells grow, but the pressure is balanced. It can't get to be too big. Maybe 40 was too small a cutoff. It looks like my pressure is always bigger than 40. So let me pick a bigger number. Well, it divided, but much more slowly. So why don't people try that out? So what did we do? We said instead of having the cell grow at a constant rate, it grows at a rate that is 1 minus the pressure divided by 40. And to make sure that the cell doesn't shrink if the pressure is large, I maxed that rate with 0. So the cell doesn't shrink. And you'll notice now cells are growing, but the growth is a lot more controlled than it was before. And when the cells run out of room and bump into the boundary, it'll fill all of the available space and then stop growing. Notice that the maximum pressure here is 70, 78, something like that. It's more than 40, but before the pressure was going up in the hundreds. And this is not a terrible model of tissue recovery. In other words, if I grow this until the cells stop dividing because they're under pressure, and then I killed a bunch of cells in the middle by deleting them, the cells next to them would start proliferating again, and they would grow until they reform the tissue. And so this would actually not be a terrible model. It's not a great model, but it's not a terrible model for wound healing or for normal tissues. One of the problems in tumors is that cells don't respect the contact inhibition of proliferation, and so they keep growing. And one of the consequences of that is that pressures inside of tumors tend to be quite high. So let's see what happens if this stops. Number of cells, 2,700 here. Let's see what happens when it fills in that extra space in the corners. Squeeze that blue out of existence. 2,819, 2,828, 2,837. Still going up a little bit. But if I wait, I will get to a situation eventually where the pressures are equilibrated and I don't have any more cell growth. Yeah, now 45, 46, 46. Have to be a little patient. Okay, so I appreciate everybody's patience with my typos. I hope that gives you a little bit of an experience for modeling cell growth and division and what it looks like in CompuCell. Again, we had to do a little bit more to implement cell division and cell growth than we did in the things we were doing before, but it's not really that bad. Again, conceptually, we create mitosis steppable. We create a placeholder cells to divide, which is a list. We check a condition on the cells that we want to divide. When we have the cells divide, we call self-divide cells with the orientation rule that we want. And then we use this update attributes to identify the volume, target volume for the new cells. We clone the properties of the parent to the child. And then if we want, we could change the child properties. For example, changing the type of the parent of the daughter cells. And so it's really only about 6 lines of code to implement cell division. Now, the rules that you use in terms of the conditions and what happens after cell division could be fairly baroque, but the basic concept is not too bad. And so here I've gone back to the one where the cell type changes. So I hope that made sense for people. I appreciate your patience. So as usual, we don't know exactly how far we'll get. So Josh and I will type up a couple of exercises to explore cell division and growth for next week. But the primary assignment for next week is to plan your project. And I hope everybody is okay. And I'll see you next week if there are no more questions.